Den er god.
J'étais ici hier pour planifier ce matin parce que chaque lundi à 8 heures, il faut être...
Oui, absolument. Alors, par contre, je vais éteindre la lumière.
Ok. Oh oui, oui.
Voilà. Alors, il faut d'abord tout éteindre.
Voilà.
Pour ensuite me rallumer le numéro.
Ja, blir det klart?
Ok. Très bien. Tout à l'heure.
Da blir det i hvert fall etter sånn på undergrep. Da får vi veldig god trening i akkurat det der. November og desember og januar er fryktelig mørke, kan du si. Spesielt desember, for da er det slutten av november. Nei, jeg synes det var fint, men du må liksom komme deg til klokka, nå vet jeg klokka 8. Så jeg må stå uansett hvordan jeg er ute.
Og så Tone, ja, så skrev jeg, ja, men det var en god idé, ja, da er jeg veldig interessert. Så hvis du kan gjøre det. Og så fikk jeg mye til det å gjøre. Ja. Nei, dette var en kjempeerklæring. Du er nødt til å være klarere i verden. Så først sier du at
kanskje det sånn at en bok på hvert fag, og ikke bare en med for alle.
Det kan jo hende i det.
Jeg gidder aldri se på saker, for jeg vet hvor de står i boka.
Jeg bruker som å pisse ut meg, og så skrev jeg bare beklagelse, uklar, men jeg ser ikke for meg at jeg ser ikke hvor tiårig prosjektet jeg skal være middeldagen på. Så jeg kommer til å ha mye obligatorisk på våren uansett. Men hvis det er mulig å ta den på sommeren eller høsten, så kunne jeg ikke sette det. Jeg synes så bare ikke det er gøy.
Hallå! Var det hyggligt med besök? Hæ? Ja, jeg trodde det skulle være 2 timer, men det var jo 5. Nei. Du kommer til å... Det er jo en ting. Det er jo stykker å gå på ja. That's hard because you guys made a number. Aren't you the introduction?
Oh, yes.
Yeah.
So,
Well, it's going to be hard. But who else is going to be Group 5. You were which group were you? 1 or 5? 5. No. Let me see if I can find group 1.
Do we have to discuss?
Okay, it's Monday morning and we're feeling it. And of course it's Monday and Chris just informed me that parts of the group 5 who were going to do the introduction are stuck in a traffic jam. So there's another group that was going to do the introduction, group 1, and I can't find your file, but that doesn't mean you didn't submit it. So group 1, while I'm doing the introductory talk, can you come and load up your slides, please, somehow or other? Right? Did you send it to me?
No, I think I uploaded it.
I can't find it, because at least you can't find it there.
But we have it in our...
Okay, okay. Good. And because it's Monday, by the way, okay, last week we did that, the article with Stop It. As soon as those guys are ready, we're ready to go. And we did the article with Stapa, and we've done the articles with Weir and with Puri and Stannard. Have you started to change your mind about what scientific articles are about? Have you started to lose your fear and awe? Yeah, good, good. Because it's building up to put you in the context. And I know that since a lot of you have not actually used the scientific literature before now, which I think is unfortunate, it really means you need a crash course in this, because you need to use the scientific literature for your own theses and to put your own work into context. But you also need to be able to use it functionally. Like, it's not just take out a few sentences and say this is correct. You have to put it into a good context. And you might think that the more references you've got, the better it is and so on. Not necessarily. And it's not always that, say, 10 articles agree with each other. They've often got different angles on things and so on. And they've very often had other things going on. And the point being that none of these articles is written all by itself. They're the result of a piece of work that has been happening in the real world, usually, unless it's a review article. And those things that have been happening in the real world have been in context with something else that's going on that's much bigger than just a little classroom project. And even though it is really easy to go down and focus on a detail, which is what you guys have done the exercise on, it's also very important to take the step out and take a look at what's happening around. So, I don't know if you guys are ready? Yeah? Okay. However you get it up, I'll just keep blathering along. Let me know when you're ready. So that when you look at where the work has been done, I mean, those 21 questions that you have, those have been refined over the years so that we know that you can use them and basically analyze anything with them and sort of at least get a little warning sign in your head that something odd is going on. But it also helps you to look at the quality of the articles and just sort of skim for what are the important points. And very often, the important points are in the materials and methods. And really, the devil is in the details. But some of the consequences that come out of that are often hidden until you read the results and the discussion. So it might look like materials and methods is good or bad, and then when you look at it in the context of results and discussion, then it starts to fall apart. And especially if you start to look at how they wanted to approach the topic. Now, since you've all read this one, of course I have a purpose with this one. And it's different again. So I'll take my phone and leave it over to you guys. And since, well, someplace I have my phone. There it is. There it is. And leave it over to you guys. And then when we're done with this, I want a discussion with you all about what you feel you have learned from these last couple of days, because we're going to keep going on Friday. So, oh, you made it! Yeah. I got the message that you were stuck in a traffic jam.
Yeah.
Yeah, not anymore.
Here I am.
Okay, good. Right. I'll move myself over here. Whenever you're ready, I suppose.
We have the article by Vike et al. It's about ISA virus in Chile. ISA virus is infection salmon anemia, and it's very common in all countries where salmon is found, and it's very common also in fish farming, and it's been seen in Norway that it's been transmitted a lot. And what they've done is they looked at the different salmon genes and the genes of this virus to see where it comes from. And recently they found this virus in Chile as well. And that's because Chile imports all their fish eggs, salmon eggs from Norway, and now they're looking to see if there is vertical transmission in Chile as well.
And what does vertical transmission mean?
That the virus jumps from populations.
Nope.
Nope.
It means going from mother to offspring.
Okay.
So it's transmitted through the generations. Horizontal is going between populations or between farms. So vertical transmission means the brood stock is giving the eggs the virus.
Okay. So does the introduction provide clearly the rationale for the study? Not very clearly, I think, but does say, well, that they found these segments and that they're comparing them to known ISI virus from Norway and the Northern Hemisphere. There's not really a very clearly stated hypothesis, but it does say what they're going to do, that they're going to yeah, they say that they're just going to present the partial genome sequences of the virus and yeah, at the seaside of ISA in Chile. So has the research question been answered previously by another study? This article refers to 2 other studies that have shown vertical transmission of ISA virus, but there are some differences. They also say that there are no studies documenting transmission between farming sites, but we don't know, maybe there are other articles that they haven't, other studies that they haven't referred to, I don't know.
So, in conclusion, if the introduction is well written or it is logical to understand, generally, yes. We think that it gives them kind of a small introduction to what they're going to research on, but there is not too many references in the introduction. And also in the end of the introduction, there's a lot of references to work done by one of the authors. And we think that maybe using a lot of self-citing can be good if the facts is true. But if the facts aren't true, then maybe it can be a little bit misleading direction in the introduction. Yes. So our conclusion is that it is okay, but it needs to be a little bit reviewed. Then it would be better.
Okay, so as I've understood what you were saying, the paper was done because, why was the paper done again?
It wasn't very clearly stated.
It wasn't very clearly stated. Okay. But because there's been outbreaks of ISA in Chile, where they're farming Atlantic salmon, but where their natural populations are Pacific salmon. And they wanted to know where the virus comes from. And it's what you said, it's very common. Yes, it's common in seawater, but it doesn't always cause diseases. But in this case it was, wasn't it? Was it causing disease in Chile? Was it causing mortalities?
I think it was.
Mm-hmm.
Yeah.
Okay. Right. So anybody else have any other questions? Do you guys have something you want to put up? I love you're on the screen. You're up and away. But if you want to do that.
Okay. Yeah. I found that there's a rationale for the study. So based on the article, my understanding is that they wanted to isolate the virus that's infecting the fish in Chile, and they want to use genetic analysis to compare with virus from other countries, known virus from other countries, and therefore to determine what type of virus it is and where it comes from.
Right.
So kind of like you just said. So.
Yeah.
But yeah, I agree that the hypothesis is not clearly stated. They did mention in the introduction about the speculation about the origin of the virus, because they knew that Norway has been exporting embryos.
Eggs. Yeah.
Yeah. so then they're kind of like making that connection that perhaps the virus that's found in Chile originates from Norway.
Right.
And later on in the study.
Yeah. Okay. Anybody have anything else to ask or add? Okay. Thanks. Okay. And then I loaded up 2 materials and methods groups. Let me see which one I get first here. Okay, this one, group 6. Can you come and do this one?
We're missing one person, but
Can you do it anyway?
I guess we could do it. Is it okay?
Let's just wing it.
Okay, what's Yanu supposed to do?
Anyway, just ignore that. So we're talking about the materials and methods for the article that was just mentioned for the introduction. And the method was briefly summarized that the eggs originating from Norway are exported to Chile, and the study was based on the seawater adult salmon from the marine farms and the freshwater juveniles. And they saw that both the juveniles and the adults have the ISA virus, but it's only the adults in the seawater that have the outbreak of the ISA disease. So what they wanted to do is to test if the ISA virus strains found in the adults and the juveniles have been vertically transmitted.
Okay. And they basically isolated the virus from the salmon and cultivated it in salmon kidney cells in cell culture. And then they extracted the RNA of the virus and applied a reverse transcription PCR to gain DNA. And this DNA was then, missing a word here, amplified in PCR and targeted with different primers to obtain 3 different genes. And after the PCR, the DNA was sequenced.
Yep. And then after they assembled the data using appropriate software, as you can see in the bottom right picture, and they identified the sequences with GeneBank, the online source, if they're any identifiable, and then they were aligned. So then they performed pairwise comparisons between the sequences. You didn't mention that. 2, 5, and 6. For some reason, they had all these sequences from the adults, but only 6 in the juveniles, which we don't know why. And then they built phylogenetic trees based on the analysis of these 3 genes. They used a program called Tree Puzzle to construct various phylogenetic trees. And then here's where the statistical part comes in. They used maximum likelihood statistic as well as 8-category gamma distribution to explain any gene substitutions like insertions, deletions, and yeah, anything like that. And then finally, trees are drawn with tree view, it's called. Okay. So, comments on this?
So for the materials and methods, from our point of view, we think it's clearly described and be reproduced again. Techniques and computer programs used are clearly referenced, so you can just get the references and have a view about the details of every single technique. Programs work on statistical basis, so statistical analysis was conducted. But one problem is that they didn't stated why they used 3 segments in order to compare between the type of viruses. They used 3 segments in the seawater samples, but only 1 segment in freshwater sample, which is segment number 6. And I think this make this study a little bit weak, because if you want to compare in the perspective of phylogeny, you have to use a lot of gene markers, so you can get everything. I mean, clearer. Maybe elaborate more on how virus was cultivated in ASK cells, I mean the kidney cells, because they didn't give details about how they did this in the cells, how they collect the cells, or if they inject and then dissect the cells, but no details about this. One virus isolated per site. It's only one virus, so there's no statistical analysis for this. Since contact information is given, open questions could easily be dealt with. So you can just contact the authors of the paper and ask if anything is not understood or not clear. So,
Yeah, you can do it. It's just quick. So this was, so we'll just answer to all the
so the answer for the questions is: the study well designed with the appropriate methodology? Can the methods section be used as a guide to replicate the study? Do the methods adequately test the hypothesis and are the statistics completely described? We would say yes, but I would say with some reservations.
Yeah. It's not perfect.
No, okay, but you probably said this, but what exactly did they sample? What did they sample?
What do you mean? Yeah, they didn't really state it. They just said they
Was it
took
eggs,
the
was it
virus
fish, was it, where did they get it? Which stages and how did they get it?
They just mentioned we took the virus propagated in liver cells.
Yeah.
Somehow.
From what?
They didn't say that.
Okay, there's another group that has the materials and methods, yes?
We didn't, but I know the answer.
And it is?
They collect the virus from the smolt stage in seawater, and some farm in seawater and freshwater. But they say that in freshwater there are no signs of virus, but they still collect the virus from the freshwater smolt.
There are viruses, just not outbreak. There's no disease. So.
Okay, and you guys had the other materials and methods, didn't you? Yeah?
Yeah. They don't say anything about which farm the seawater outside is from.
But is it Norway or it's Chile, yeah?
It just said Chile.
Okay, yeah.
And same for the freshwater smolt which they collected. They don't say anything about how many samples were taken.
Right. And is anything taken from the broodstock in Norway?
As far as I could understand, no.
Yes.
That's what I think is weird, because they say for the smolts, they talk about them as their brood fish. Because they, we took it from the brood fish, but they mean smolts.
Right.
And that can be confusing.
Yeah, it is. It is. Yeah. Okay, so basically we're not clear about what they sampled.
But that's not stated in the materials and methods, which it should.
Right.
Yeah.
Right. Okay. We're not clear about what they sampled or where they sampled it from.
Yeah. The only things that were clearly stated were like stuff you can't really bullshit, like statistical analysis.
Right. Oh, Stoppel bullshit statistical analysis.
I guess so.
55 papers worth.
Yeah, but all the sequencing and stuff.
Right.
I mean, that was
real. Yes. 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 Okay. So we're just pointing out the unclarity here.
Yeah.
So okay. Good. Thank you. Okay, and then the next group that was doing the results. Let's see, we have 3 and 7. Which one of you wants to go? I have 3 right here. Does group 3 want to come? Okay.
virus in Chile, evidence of vertical transmission. Do you want to go? So we found it quite complex. They basically took this whole list of every single place in Norway and the US and Chile and I think Canada on this long spreadsheet that goes on for 2 pages worth. And it's talking about the sequences from 2, 6, and 5. And it can be a bit confusing because the order that they list them in is, you know, they say 2, 6, 2, 5, and 6, but they've actually listed it as 2, 6, 5, so it can be kind of misrepresented if you don't read it closely enough. Plus the table legend, it's half of it's above the table and half of it's below the table, so it's difficult. You have to keep jumping back and forth if you want the full description of it. And the figures for 2 and 3 were actually in the discussion section, not in the results. So you have to go really far down if they reference it in the text. And then for table 1, there was no reference to this table at all, anywhere in the results. So it should basically be an appendix section, especially because it is this complex and it provides too much information to read in the results. And yeah, it's the inconsistent labeling as well, like as I just mentioned.
We don't mention anything about if it's one individual, each segment, or anything about like the details in the study.
Yeah, they just said these are the sequences that we found, and these are the places, and that's it. So not very clear, and a little overinterpreted for this table at least.
And we think that it maybe should go in the appendix instead and just take the most important small parts of the table and not the whole big information.
And explain what the sequences mean.
Yeah.
Right.
Is the collecting of data sufficiently described? We don't think so. It's only mentioned to phylogenetic analysis and blast search,
but not how it's used or anything like that.
And we didn't really, if it's biased or small, we don't really know because of the information given, and it's hard to determine if it's big or small since they have not much.
They didn't say how many different sequences they were doing or anything. So.
And since they only taken those 3 segments as well, it can be a lot bigger, but they only they made it smaller. So yeah.
And the figures and tables illustrate important features of methods and results. We think they should simplify like the phylogenetic tree, especially this one, because it's all about the sequences and how they're labeled and what they go to, but it's really confusing if you don't understand like the whole genetics behind it and the labeling that they use. They don't explain it very well either. And the legend is kind of on the side, which was also kind of confusing because it's supposed to be on the bottom, underneath. So it just looks kind of compressed in a lot of information in one area, we thought. Anything to add? Yeah, we had general, did you want discussion?
No, actually I wanted to know what they found.
Okay.
Because that's part of the results. What do they say they found?
Well, basically, they found that the samples that it took from Norway had the virus, and that it's actually that it's not existent in Chile previously. So that there is a link from Norway fish to the ones in Chile, and that it's actually from there. And because it's not natural in Chile as its own virus, it never existed there, so we've kind of introduced it through fish in Norway.
So they're saying that.
Yeah.
Okay. So they're saying that just by process of elimination, it has to have been imported to Chile.
Yeah, basically. Yeah.
And the other results group, where are you? And do you want to add anything?
Well, we're gonna add.
Well, we at least have one sentence from the results part we find quite interesting, because we were talking about taking segments from different numbers or stocks or whatever, and suddenly they say the other segments from blah blah were not sequenced, and they give no answer for why they don't sequence that part. And then suddenly as a reader, like, okay, why do they not give an answer? Like, are they starting to just kind of find the results they want,
right,
and kind of stop there? Not like, yeah. some red lamps are starting to blink in the horizon.
Right. Yeah.
And since they all already says it like they write it were not sequence, it should at least be mentioned why.
Yeah.
If they have a good reason.
Or even how many other sequences are possible. Well, there's quite a lot, but
yeah.
Yeah. Yeah. Okay.
And also we do agree with the tables, because the one you mentioned that's actually for the materials and methods and are listed there. So why the heck do it comes in the result part?
Yeah. Yeah.
Also, it's hard as a reader to follow sometimes, and there's a lot of shortings of words.
Yeah,
and when
abbreviations, yes.
when you're not in this business, you kind of fall off, to be honest.
Yeah.
It's possible to understand.
Yeah. Yeah.
Pretty much.
Right. Okay.
So
So.
kind of as well when we searched up the paper and it got some kind of into a critical trial or something.
Right.
For was it falsifying?
I think so.
Oh yeah, but we'll get to that.
Yeah,
We'll get to that.
Yeah.
Yeah. Yeah. Right.
Questions about that.
Okay. Yeah, because there's quite a lot to say.
Yeah.
Right. Okay. Anything else you want to add?
We had a general section, but did you want discussion?
Yeah, we'll go discussion first. Yeah. Okay. Good. Thank you. And then we have. Yeah, there it was. 4, 8, and 9. Which one of you wants to come up? I have loaded up 4 and 9 and oh, look at that! I have all 3 right there. Which one of you wants to come first? Since you're standing, which one are you? Oh, sorry. Okay, come on. Either 4, 8, or 9 comes up. I'm making a discussion. Oh, that's the introduction.
So then,
okay, come on, group 8. There we go. Group 8, you have been volunteered. Where the heck is it?
okay, we summarize a little bit about the article. The phylogenetic analysis comparing the likelihood between genome, focusing on species specific segments of a virus isolated from SMART and for-SMART in Chile and those from EU and North America. The author conclude about the origin of the virus in Chile is from Norway by vertical transmission. And this is summarize how the reason for the papers that some other groups are also saying about that. So I don't want to go in detail about this.
Okay, so there are some. I will generally say that I think the whole article is quite well written, and they have make some good points and have some good ideas for what. But however, there are some things that could be criticized, like they don't initially start discussion results, and, or it's not very clear that they do, and yeah, they're not being clear when discussing who says what and where the information is coming from. So yeah, they should.
They just start to talk about things and not referencing where the information is coming from, and they don't use actual references in the whole first part of the discussion.
Yeah, and they kind of lack like critical evaluation of their own work. It seems like, and to draw that conclusion that it is vertical transmission, they should have included some other factors as well, because it seems like it's vertical transmission, and it probably is, but yeah, they should have also included some other things, I would say, and it seems like they're also lack the critical evaluation of their own results, and that they're only using references that support their hypothesis. Yeah, and yeah, the materials and methods that have been used are not sufficient to allow the conclusion. And yeah.
Because they draw a conclusion in the discussion, but
Yeah, and that conclusion is?
That it is vertically transmitted
right.
from Norway.
Yeah.
Or yeah.
But when you go back and look at the materials and methods and the discussion and the results together, you don't feel convinced about the
they should have said like
information that they give.
It probably is, or it could be vertical transmission, but this is definitely vertical transmission.
Right.
Yeah.
And there are, this is maybe their horizontal or vertical. They didn't consider that it may somehow, perhaps it happen or not. And then I saw in the introduction in material method, they said about Chile was imported embryos from USA and Norway, but there's a slate from US that called the Maine. Otherwise, they also use the slate from North America, but also there are another slates from America they call Maine. They didn't use that. Why they didn't use? They don't mention.
Possibly sales reasons.
Yeah. And there are a lot of factors that may affect the infectious virus. For example, the stage of the fish and other things they didn't mention.
Yeah.
And we also found some articles said about this case that there's a committee talk about that. Did they cheating or not with this article?
We're going to go into that quite a lot.
Okay.
Yeah.
And then we didn't say that they cheating or like that.
Right.
Yeah.
Right. Okay.
This is we answered the question. That the research question really clearly stated and unexpected findings.
It's answered. It's not stated at the beginning with, but they tell them like as what they have shown with their paper after a while. But you didn't really know that's what they were after in the first place.
Right.
So,
Unexpected findings. No. Limitation addressed. No, that's what I'm talking before. And reasonable claim that, like we said before, that no some part that no citation, and some point yes, but there are another comments.
well, yeah. So just based on that article, we found that they have been accused of scientific cheating.
Wow.
We think like the science is probably right, and they probably knew it was right, but I think that has biased the way they wrote the report because they don't really back up what they're saying with like appropriate information and appropriate tests. It's like they decided when they sat down and wrote it that this is the outcome. And
They might have a preferred explanation for their hypothesis.
Yeah. Some.
Yeah.
Yeah.
That could
Okay.
affect how they.
Right.
Yeah.
Right. Okay. Anybody else want to add anything? Yes.
Yeah. Groups 9 have some
louder.
Yeah. Because my group also study some discussion about this article. So I have a quite different look about this article in discussion. Because I think hypothesis of this article is try to find where how to say if ISA is transmitted vertical from Norway to another country to Chile by exporting infected embryo or not, and when we look at discussion, firstly, I think that her hypothesis quite clearly, and result and discussion quite clearly. I can support some information.
Okay, so you
why
your group liked it better.
Yeah,
your group thought it was better constructed. Okay.
I think results show that ISA found in Chilean fish farming is close to Norwegian genotype, and we can sort in this figure one, and we based on phylogenetic tree, we can say that
Chile's in the middle of the Norwegian race.
It's very close. It's similar to Norway, most of them, and far away from genotype North America.
Right.
And more supported, we can find in figure 2 and figure 3 another segment. Is that Chile 2008 is very close to Norway and Scotland.
Right.
I mean Europeans.
Figures 2 and 3.
And so I think there is a reason why they think that maybe Chile ESA from Chile come from Norway.
Right.
Yeah, come from Norway, come from European country, and support more evidence so that ESA is absent in Oncorhynchus, another kind.
The Pacific
Salmon. Yes. Specific species in the west coast of North Africa and North America, and no natural population of salmon virus before in China, and documentation that ESA virus can be transmitted vertical from Norwegian broodstock fish in 1997. So all of information they support in discussion maybe results, maybe lead to conclusion that maybe ESA in China come from Norway. But when we look at about material and method, we can see that they collect sample from Chile, from outbreak of Chile, but we didn't collect sample from embryo and broodstock in Norway at the same time.
Right.
Yes. So maybe it's not enough evidence to support that conclusion, because at the same time maybe gene from broodstock and gene from Rio maybe a little bit different. From previous study. So I think they have a reason to believe it.
Yes.
But not enough. Not enough information.
Okay. In fact, this was just perfect. This difference between your this group's opinion and this group's opinion, because they're both justified and lead up into what we're going to be doing both the rest of today and on Friday, and this kind of how things get interpreted and whether or not you agree is key to what happened to this paper. I'm just checking the time. Okay. A 10-minute break. We're going to come back, discuss a little bit of what you've learned, and then go into the real nitty-gritty stuff, because now the fun begins. Okay, and oh, very good. 3 stars. Good. Yes. Okay. So 7-minute break, and then we'll be back. Oh, I have. Oh.
You want me to just edit that? Okay, let me just edit it.
Ja, det er sånn. Ja.
Are we missing anybody? A little bit. Okay. So basically, what I want to know is what you've learned from this last little section about the critical reading, from when we started the first article to the last. And I know it's really hard to sort of ask somebody straight up, so I'll give you like a minute to talk with the people beside you and come up with something that I can use. And just let me know what your experiences have been. If you've been surprised, if you've been, if you've learned some strategies, just think about these kind of things. What have you learned? What can you apply from this now? So I'll give you a minute.
Den siste var jo litt bare spesielt. Du må jo virkelig bare sønge det. Du må virkelig bare sønge det. Først var det bare det. Hva går det? Hva var det?
Okay, winding up. I see a lot of smiles on your faces, so this was not a painful exercise. Okay, starting at the back, I guess. Tell me what you learned. Ingrid.
I've learned that I can just see where I find the article to know if I trust it or not.
Source is not equal to trust.
Yeah.
Yep. Okay, I'll just go down this side and then I'll go down this side. Yeah?
Don't trust anyone. Yeah. Well, also we need to be more critical because I've just been thinking, oh, it's published in Science, that's all right.
Right. Okay. But if you hadn't had the questions, would you have been able to be critical?
No, definitely not.
Must use questions. Okay. Give me, for example.
I think more to look more at the method and see the context from the method and what the conclusion of the paper.
Yeah. Okay, Winnie, for example. Good morning.
Pretty much things have to make sense.
Okay.
People have to make sense. References have to make sense.
Okay.
Yeah.
And before this, did you think that you didn't have the scientific background in order to detect whether or not it was sensible? There's some nods. Did you think you had, without the questions and without this kind of exercise, do you feel confident that you would have been able to detect problems?
No.
Right. And now you know it works? You feel more confident? Very good. Very good. Because, by the way, these are the exact same questions I'm going to be applying to your reports. So, okay. Yeah? No, behind you.
We talked a bit about that guy on Friday that they presented an article from about these statistic variables where every data from a decade was falsified.
Right.
And like, don't trust anything. So
yeah,
that was what we discussed.
Right. Yeah, I know, that one's a real eye-opener. I couldn't have made up a better story than that one, you know, and it's so well documented that it was a surprise to find it. It was a joy to find it because it made the point so clearly. Yeah, you guys? Say good?
Well, I guess Google is your friend sometimes. But yeah, Google the authors, because I think most of the articles we've read, you'll find something on Google that's, I mean, not always you'll find that, but if it's something very critical, someone else might have noticed.
Right, yeah. And sometimes Retraction Watch is a very good place to go. Yeah. Okay.
I think we need support enough literature relevant to our research question. I mean.
Okay, so you need more than just the author's self-citation. You need more than.
No, I mean we need put more literature.
Okay, so you need more exercise in examining literature. Is that what you mean? Or you need more background to examine the literature?
No. Reference? Reference, yes.
Ah!
I mean.
I'm going to... I'm going to write it like here, need to link papers findings with other literature. And that's, okay, even though this might not be exactly what you meant, this is a very good point. Because when you are now going to be writing your discussion, because this week I'll get through your results, when you are going to be writing your discussion, you're going to be finding literature that supports one thing and then finding literature that supports the exact opposite. And so that's why this part here and the critical examination of the weaknesses and so on is very important in using the literature, knowing whether or not you got a good article or a bad article to support what you found is quite critical because you're going to be building on it. Okay. How about you guys at the back there? Hannah, Kat?
Well, I guess we were all also discussing how with Vicky et al., they were also self-citing. It's not necessarily a bad thing.
No, it's not.
But I mean, because you've been doing kind of this research your whole academic career, so.
Right.
But as long as you kind of make sure that you should be skeptical about that. So.
Right. Okay. You've had experience in critical reading. Anything new you learned?
Me?
Yeah.
Yeah, you sort of always when you read something and you sort of buy it, and then you find out later that after picking at it and after hearing what other people say that actually it's not what it all seems to be. So you sort of learn to doubt yourself as well when you
right. That's actually a good point. I'll say first impressions. Okay. Hassan?
Yeah, I was thinking when I used to read some kind of articles and I just found it complicated. I was saying the fault is me. I mean, it was my fault because I couldn't understand the paper and I just keep reading it again and again and again. But now I would say that every single paper that's published should be at least 3 for the relevant people. Yeah, so everyone who tries to read this paper should be able to understand what they are mean. Another thing, maybe if you could apply the review for each article. I mean the review for the article would be beneficial because you can at least know the impression of from other people, what they found in the article, what they said about the article.
Right.
Yeah.
Okay. So should reviewers' comments be available?
Yes. Maybe you can just modify the question, the question that we tested, I mean the paper, to be just fit with the kind of study you want to do.
I'm sorry, I didn't understand that.
I mean the question that we applied to the paper to check it. Maybe we can modify it a little bit just to fit the study you intend to do. Be modified.
Yeah, for example, the question is a hypothesis clearly stated. Isn't hypothesis always really necessary to a paper?
Actually, it's not. Yeah. Right. So, Marek, did you have any more to add?
Yeah, I would say basically be careful with the conclusions. Always, because that's what we are going to conclude, and that's what we are going to cite also.
Yeah,
because
very good. Very good.
I would say conclude what you have done.
Yeah.
Because especially with the weak setting, everything was perfect.
Yeah.
The conclusion was quite exaggerated. They have concluded something, but still there is some gap to conclude that.
Right.
So conclusions matter.
They do. They matter a great deal. Yes.
They make a very absolute conclusion.
Right.
So is that why make them in trouble with that?
Right. I'm going to put this in big letters, actually. Because often the conclusions and the abstract is all that gets read. And when you're searching for papers, you go by title and keywords. And then you sort by what the abstract says. And usually, if you're in a rush for time, you go title, abstract, blah, 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 conclusion. Yeah. And that means that what you write in conclusions and what you read in conclusions should be aligned with what the rest of the paper is, but you know now it is not. So if those conclusions are relevant for your work, very relevant, go back and check the paper. Make sure that that paper's materials and methods and the context and the results actually support this conclusion. It's really key. How about you guys?
Well, my first thought was that we should do more than just read the abstract.
Yeah. Read the whole thing.
And I wish that would be more prestige in replicating studies and look for bad science and so on.
Yeah.
Because it's getting almost as important as doing science is to find bad science. So.
Yeah, I agree. I agree. Yeah. Justin, Chris?
Yeah, I learned how to characterize a paper scientifically, and maybe we should have a serious attitude towards our own data in the future.
Yes, very good. Especially since I'm going to be grading you on it. But yes, a serious attitude to your own data. Because these kind of questions, these 21 questions that I gave you, they are the ones that reviewers generally use on a manuscript, if the reviewers are good. But of course, they're not always good. And obviously, lots of stuff gets through the so-called peer review process. And I'll be coming back to the peer review process later. Would you have felt confident trying to write a paper like Vika et al. Without, say, a little course in how to write the IMRAD, the Introduction, Materials, Methods, Results, and Discussion? Would you have felt confident? No.
No.
Right. Okay. This is one of the things here. This paper was the first one that Siri ever wrote, ever, in the IMRAD fashion, in the Introduction, Materials, Methods, Results, and Discussion. And she had had no training in this. And yes, you found a little bit about the authors online, but it was, and the discussion is, or the fallout from this paper, it looks immediately, from close up, it looks like it's fraud, not fraud. That's the discussion. But in fact, the shit hit the fan globally. And what was really interesting, and the reason why I built this up, is because, yes, there's a little thing around fraud, not fraud, but the impact of this paper went literally around the globe and is still going on. But it's partly because of the paper, partly because of the fuss and focus, partly because of the economic impact of what it actually said. And this is why it's like, it's more fun. Okay, when some of you found, you said, oh, it was accused of fraud. No, nothing. The emotion and the physicality, the arguments, the long-term accusations was just phenomenal. And that's what's the part of the next exercise. Because, let's see here. Okay, I have now loaded up. Let me see if it'll show here. Come on, maybe it's there. I wrote it up, ethical role play assignment. Okay, And let me say open. Let's see if I can get both pages out from this. Yeah, there we go. Can you read that? No.
No.
Okay. I'll read it for you so you recognize it anyway. Let me see. Can I do this better? There we go. Okay. Because one of the things here is that you have to be able to see these things in context. So when I opened this up today, I said all papers, you know, they look like they're done in isolation, but they're part of a project and that's part of a response to a real world situation. And here, paragraph 3 here, it says, oh, paragraph 2 there. Critical evaluation is not limited to detecting cases of poor experimental design, plagiarism, or fraudulent data. Most articles are done in a bigger world context, and the impact of the written word plays out long after publication. And in the case of international industries, which aquaculture is, based on internationally traded biological resources, this can be especially tricky, okay? So what we're going to do is go through some roles here. And you're going to get the chance to prepare it, because the shit really, really did hit the fan. And you're going to be taking groups. Now, these groups, I'm going to be handing out, and I put the papers over there. Okay. So, this is basically, let me see if I can do this better. Oops. Come on. There. Okay. Now, see, there's 9 roles. Actually, there's more if I want to split it up. And the point is that, let's see, here we go. The point is that it went very far. Role number 2, Aquagen, it's a real company. They're the ones that sent the letter about scientific fraud. They started the ball rolling. But that's also because they were one of the main ones exporting eggs to Chile. The ethics committee handled the fraud case, and one of you, group number 3 is going to be the ethics committee, and just sort of be themselves. Group number 4 is Cermak, and Cermak was Vika's employer at the time. And then the Cohen Commission, they were in Canada, in British Columbia, where there's huge runs of Pacific salmon, and there's lots and lots of fights between the wild and farm salmon groups there. The federally appointed Cohen Commission was set up to look at, scientifically, the interaction between wild and farm salmon, and they had a special session on infectious salmon anemia. You can even see it online. And okay, this Cohen Commission, it worked for over a year and a half, hearing people, gathering evidence and so on, and they had the Prime Minister's office stamp, you can collect whatever evidence from whatever person you want. And they had thousands of people coming in and giving evidence one way or the other, but a special session on infectious salmon anemia. And then you have the environmental groups that I just mentioned. You know, there's one group here that's quite vociferous and so on, and some of you will recognize who it could possibly be. And there was another group over there in Canada called, with a guy called Don Staniford, who was who's been in court many times. And group number 6 gets that. They also get some of these real, where's group number 6? There you are, okay. You get some of these real placards that they were spreading around that look like cigarette packages. I got them from Don Staniford. And it says things like, Norwegian-owned farm salmon is pure aquatic terrorism. And salmon farms are cancer, and things like that, okay? Right, okay? And they joined forces with the group over here. And that's, you know, you can pass these along, but hand them back to me. And I want those back too, because these are my only copies now. So, and there's quite a variety of those things. So this was how far it spread. And it also spread into a movie that was playing at BIF, what was it, last year? And then we have the University of Bergen. Also, the authors are employed at the University of Bergen. So group number 7 is going to be that group, and you'll be able to find what was done or not. Then the National Fisheries and Aquaculture Union's Research Fund, that's FOF. for those who know it. And they are part, they get a research tariff. There's a lot of money being made in fisheries and aquaculture, and what is it, 0.03% or 0.3% of the profit from that year's fish farming exports gets put into a research fund that is then delegated to questions that the industry itself thinks is interesting to do research on. So it's like separate from the National Research Council, and they can selectively choose what they want to focus on or not. And they have a budget of about 120 million a year, okay? So this is what they can put into problems and scientific efforts to go on. And then you have the Chilean aquaculture industry, which really suffered. So the thing is that all of these were doing their job. And okay, I'm just going to hand out these. Group number 1, where are you? Okay, these are group descriptions. And group number 2, here we are. Okay, so there we go. 3. Now stretch over to you. Okay, and I don't want you to see each other's. 4. 5. 5. There you go. Okay. 6. Okay. Let me see what we got. 7 go. 7.
Late 8 here.
Huh?
8 here.
Okay, good, thank you. Yep. And, okay, I've only got 7 and 9. Is 9 on this side?
There's 2 9s, 1 here.
Okay. There's a 9 and a 7. Okay. Okay. And now, one of the things is that facts is one thing, but how you feel is another. And that was one of the reasons why. Are those little cigarette package warnings still going around? Oh, there they are. Yeah. Yeah, just spread them out. I just want them back again, except for the ones I gave the group that's playing the environmental groups. Just spread them out and show them. Because they are really in your face. And they were really very confrontational. And when these kinds of things happened, it feels horrible. So as this goes on, okay, as this goes on, okay, let me see. You should approach your role as though you were the real people or parties involved. The more authentic and vivid and active, the more you will all learn. Because being in the eye of the storm or being this focused on is terribly disorienting. So what you're going to do is this. You're going to define for the other people, so you make us understand who you are and what you represent. And you can do it any way you want. I mean, literally, the more vivid, the more authentic you take on that role, the better it is. You know, if your group has turned out to be very sort of officious and quiet and like this, that's the way you play it. If your group is for the people, by the people, come on, you know, justice, that's the way you play it. If your group is we disagree with everything, leave the wild stocks alone, don't farm anything, that's the way you play it. If your group is, I got my career, that's the way you play it, okay? So you make us feel what that group has done. And, okay, so because this really, really makes a difference to how the whole exercise of what the impact of this can be has played out. And so basically what we want you to do is to be very convincing. And you make statements to protect your role. And in this case, it's not a Norwegian debate trying to find out where we agree or not. It is the confrontation, because it really was. And it is the bare presentation of one standpoint or another. And okay, this is how we're going to do it on Friday. So there's 2 rounds, and round 1 is the statement of who you are, and then it is round 2, where each role states how they've been impacted by the paper or by ISA. And to the degree that you have. Now, all of this information is basically online, and it dates from around when the paper came out. So if you look at all of your groups and put in the date, you'll be able to find stuff. But the Chilean industry has collapsed and has reformed and so on. Which one has the Chilean industry? Okay, yeah, because that has collapsed and reformed. Find out something about the history of it, because literally it impacted on daily life in Chile. It really did. And so, yes?
Can I just ask you a question about people writing this?
Yep.
Were they doctorants or?
Professor.
She was trying to get her PhD.
She was trying to get her PhD.
Yeah.
So shouldn't, writing such a paper that has such large economic or could have consequences, wouldn't she somehow be more prepared for that by her professors and by her, by the institution? Or she never written a paper in her life. It's a very massive project to actually go out and actually say something like that towards an industry.
Right.
It's $1 million industry. So
It's $1 billion industry. Yeah.
Industry.
Yeah.
And it's quite, I don't know, I feel it's very immature, the whole process and the writing of the report.
Right. And
she should know what she's taking on, kind of.
And I am more curious about the reviewers.
Supervisor should be.
Actually, the reviewers' comments I don't have, but I'm sure I could get them dug up. Very good point. And in fact, Siri, Vika, she helped me develop this exercise. So this is not going behind her back. I got a lot of the information from her, and at one point I had the original letter that accused them of fraud, so you could actually see the text. And it might be online, and I might find it between now and Friday. You got it?
I just searched for the article, and there's a link to it.
Ah, great. Okay.
And I saw a report from committee in English.
Right. Yes. 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 So, but the process. Okay. Look at the timing of this. So when you're looking at the documents, also look at the dates so that you know what happened when. And your point is very well taken. And remember at the beginning I said there are cultural differences between how you approach scientific articles. And I repeat that in the mid-80s, mid-1980s, the Norwegian authorities and the Institute of Marine Research and so on had no tradition of requiring that results be published in peer-reviewed scientific articles. And I was here then, and I arranged that some friends of mine who were on the editorial board of a well-regarded journal, the Canadian Journal of Fisheries and Aquatic Sciences, they came over here and gave a short course in how to write a scientific paper to permanently employed scientists at the Institute of Marine Research. Because that's how new that culture of writing something for peer review is in Norway. And this may look, okay, now it's 30 years or something like that, but it takes time for these kind of things to seep through. And it also takes time, like we already know that within this class, there are cultural differences in your exposure to using scientific articles and to whether or not they are built up in the IMRAD fashion or not, and how often and how comfortable you look at it. And when you come to the master's level, there's also a difference here. Culturally, everybody in bio, except fish health, goes through BIO300. Because the fish health people chuck their program full of obligatory this and that's, and how to write was not part of it. So, and we knew already that with the 2-year limitation, you at least have to get the basics of how to write a thesis, which is IMRAD anyway. So, so there's that. And then, because the tradition in Norway is, relatively speaking, short, and it was also accompanied by the tradition of, peer-reviewed scientific publication means good science, and more from the same author means good scientist. Okay, stop him is all I'm going to say. It means that at one point there's been a driver to get these publications out, but there hasn't been a driver to control the quality of what comes out. And the Nobel Prize-winning couple from Trondheim, Mosok, they also have very little good to say about what's called telekant system, counting the number of publications in Norway, because it is fraught with alternate motivations, and it is fraught with the ease of cheating the system. And there will be a lecture called The System and Scamming the System. So the point being here that you have to recognize and do good science. Okay, and now you can recognize good science, more or less. And you can certainly recognize when a paper is good or bad or has some other kind of motivations on it. And then this broader context is exactly where we want to go with this, because it was phenomenal. When they were accused of fraud, the only thing they did for 2 years was defend themselves. Defend themselves. No research, no publications, no funding, and they needed a lawyer. And that lawyer is expensive. And that lawyer was paid by Cermaq, because Cermaq was technically Siri's employer. That lawyer cost a fortune. They would not, the authors themselves would never have been able to afford this. And it went back and forth and back and forth. And it involved experts, the only global experts that exist in ISA were put on this case to say whether or not it was fraud. And for those people representing the ethics committee, take careful note of what exactly is said in the conclusions, because that also is a clue for how much of a judgment, how subjective, objective this can be. And it's also the reason why I'm giving you this exercise, so you can push the other people's points of view. I mean, you guys are basically heading towards eventually writing your own first scientific publication. Not all of you, but to that. And this is why I want you to always think about the big context. Yes, Hannah?
This critical reading is directed at research articles, but what about review articles? Shouldn't we be just as critical?
Yes, yes. But they're harder to be critical about.
Yeah.
Yeah.
I mean, they're synthesizing all this.
Yes.
From so many papers.
Yes.
And it's not in the IMRAD format, so you
No.
just have a different way of looking at it.
Yes. Yeah, and review articles need to be read very carefully.
And they're very long.
Usually they are, yeah. But they're useful because, you know, with all these small publications coming out in a field, you need to say, okay, the field has moved here, how? And sort of justify and sort of set the stage again. And that's one of the reasons why we do this. Okay, so in that round 2 here, when you guys are playing your roles, and play them as hard as you want, literally. I don't care if you come in costume to give the thing reality, because it had bite, it had drama, and you should feel this. But you should feel this from each player's point of view, because that gives that context, that real world context. So there's a little bit of work to figure out what happened online, and then it is to develop your roles and show the others what your standpoint is, your group's standpoint is on Friday. And if it gets loud and noisy, all the better, because you're supposed to represent that group and feel as they felt, and the more authentic it is, the more understandable the whole process will seem. And it's quite enlightening. It's also quite shocking in a way, but it keeps going. So this is the last sort of stage in this critical reading, putting it into the real world context and seeing that big real world context, feeling it will make it be better. So, you know, those posters, have they gone around? Can I get them back?
Nope, still no.
Still no? Okay. Well, anyway, for those who want to, I'll just spread them over here, so if you want to see some. There's about 7 or 8 different ones. Oh gosh, there's more. So, yes, there's even one that says salmon farming kills workers. And that's in reference to, that's also in reference to the Chileans. Oh yeah, right, yes, there's that one too. Kills workers, our cancer. Okay, actually I've got 10 different ones here. 10 different ones. And by the way, all of them start out at the top saying Norwegian owned. And to put it mildly, the anti-aquaculture groups had a field day off of the impact of salmon mortalities and sending biological material around the world and so on. It is important to know that Atlantic salmon are one particular species that are not found in the Pacific, but it is Atlantic salmon that are farmed in the Pacific. And there's 5 different species of Pacific salmon, and they're the ones who return and go up the river and die en masse after they spawned. Atlantic salmon do not. Okay? And that's it. So I'm looking forward to seeing what happens on Friday. There's 10 different versions of the opposite pole right there. I keep hearing information in quotation marks about salmon farming or fish farming around the world and what it does. There is big opposition for all of it, but there's also people who are trying desperately to find this out. So in the Cohen Commission, oh, the Chilean farmers, I think I've got something for you. So at the end, when we can say that we're done, and you guys are now working in groups to prepare on Friday, and just enjoy this. Feel it. Put yourselves in the situation. You do not have to know what the others are thinking in order to make this a real and exciting exercise. Okay, so fall into the role, find your arguments, find out what happened, and present it here. And group 7 was. Let's see.
University of Bergen.
Yeah, exactly. Group 7, can you come and talk with me for a second? Okay, and that's it. Thank you for today.
Thank you. Hi. I have no time here for discussion, but we can discuss it on Facebook.
I'm not getting to it. I'm not actually so ask him when he calls back. As soon as Just so you know, you guys have a very because the intuition is all important individual, and this is also the problem with in general.
Always has me too.
Just like when you see a documentary or a film, you know, they're saying the views of the characters do not necessarily represent. Play it like and the Cohen Commission. Are you guys the Cohen Commission? Are you guys the Cohen Commission? Cohen Commission. I forgot the Cohen Commission because I haven't really. Oh yeah. Okay, and there's Gebel is the final report, and one of the worked for a long time. There's committee members, be those committee members, who really took it seriously, and we took the charge of fraud seriously. So I did like define what it is, define what you did, tell us what you did, and in that final report.
There we go.
You might actually read out the exact wording that clears them of fraud. Because that exact wording is important. And it's also not as exact as Yes. No. Yeah, I'll have to send everybody an email. So you can sit as groups around here. You don't have to just... Yeah, or what for example, if there was documentation, I think it's 2 years. How many people are on there, what background they have. That's quite a committee. And you have a very distanced view to us because This is the definition. This is the intent. We found that according to the definition, and put it like you would really trying to be definitionally.
Yeah, we hate.
And state how many reports you got, how long the work was, you can find out who got expert rankings from, and so on, because you did. Find out about that, and let us know the scope of the work.
But everything is this completely in one document.
Just surf the web a little bit. Yeah. But yes, in the final report, I think it's all there. The final report is the ethics committee on VK. Yeah. So you go to the conclusions, yes, but the background will detail some about how many reports came out, just summarize.
Because I think we all like the article something and they
Right. And there's, they also can get in key scientists to discuss the for and against it. Some of the findings were against it. Right. So this makes it, point that out. And I'll give it so watch that. Comes back and points him. Okay. Thank you. Which group are you? Oh, okay. Yeah, I feel like you are that.
Or someone was. Yeah, I'm a kid, so no, we'll.
Yeah, but the environment might
I don't know where even something that over that. Yeah, exactly. Just the colors. Oh, I have no problem with that. Like Hawaiian shirts, that was cool, like Hawaiian shirts. I had it in last year, but I didn't bring it home. It's too cold. Wait, are we actually like hey, you're okay. Maybe we should draw like maybe I'll buy a white t-shirt.
So you see the
Yes.
yeah,
And no.
why not? Write something on the head and then
Oh, I have all my like badges. I have all my like Harem trousers thingies, like the baggy ones. Yeah. I had 10 of those. I'll wear that. I have 3 pairs. Okay. I have purple. I have the black and the white. And I have a turquoise. I love that one. Okay. I need to watch them. I can give you the black ones. Oh, he needs colors. Yeah. I don't know. Blue, yellow.
You're also the boss.
Yeah. You are.
Oh yeah. At least I think I have a few lectures this week. This is good. Hi there.
Actually, they were working up the final defense. Literally. That was the response. Oh my God, somebody from the university.
Like this?
Did you like that? So,
When did you check that? 10 minutes ago? That's creepy.
Yeah.
How does she?
Yeah, I was like, oh, what? I saw that.
We got it off with the last sequence. With the taste and all that stuff. Yes.
So she depended on.
I think, and then I just yesterday I went to work on my report, but then I really wanted to go on the trek with Sim. I've been planning to do that, and that trek was part of the whole weekend. But one good thing came out of the weekend. I did end up being more social than this for the rest of this semester. I went to Sim twice this weekend, so thank you. Yeah, and Friday yeah, it was a lot of fun. Although I regret throwing all my big hats into that. Yeah, that was a bit silly. We saw this like fish. Exactly. Yeah, so we didn't lose. Which I'm totally into like 4 this week now. Yeah. Wow.
We've got 2 hours. Whichever one is oh my God. We'll say just public. Or something, yeah. You bring us through your experiences.